In this video, we're talking about defense against infectious disease. It's topic 6.3 and it's part 2. So if you haven't seen all the grayed out syllabus points, go back and watch part 1. Right, so far we've talked about only the non-specific lines of defense. The primary being the skin, the mucous membrane, and the chemical barriers like saliva and tears to stop foreign material entering the body. We've also talked about the secondary line of defense, which involves inflammation, so white blood cells phagocytosing foreign material and clotting pathways. We are going to now talk about the specific defenses and, more importantly, one particular type of white blood cell, which are the lymphocytes. Now, in the bloodstream, after the initial secondary defenses have completed a non-specific kind of scout, other white blood cells can produce a more specific response depending on what antigen or pathogen is available. So remember that antigen is the stimulant of the immune system, and it can be a protein on the cell surface of a bacterium or something like that, or any other cell really. So lymphocytes are a type of white blood cell which produce a protein called antibodies. Now, they are stimulated by T cells, which can see that there's other things going on in the system, and they will actually get the B cells to make this protein. Now, antibodies are short-lived proteins, and they bind to the antigen and flag it for removal by a phagocyte to engulf or kill it off by another immune cell, for example. So once the antibodies have sort of docked onto that antigen, then it can kind of flag in other cells to come and kill this or come and engulf this. All right, now B lymphocytes are made in the bone marrow, and they'll only be responsible for making one type of antibody. Only one, which is specific to one type of antigen. But there's obviously many types of these lymphocytes, so they can cover a wide range of antigens that your body might be exposed to. Once the antigen appears, it stimulates mitosis of the B cells, and some of them differentiate into plasma cells, while the other ones will differentiate into memory cells. Now, the plasma cells will completely pump out antibodies. They want to clear that infection, but these ones are short-lived. The memory cells, however, have a long lifespan, and they can recall any exposure they've had to their specific antigen, and they can become plasma cells at the next exposure. So if a new infection comes along, the lymphocytes won't initially know what antibody to produce, so they kind of just spit out whatever they can to hope it works. This is why vaccines are so important, because it's priming the immune system. It gives them an initial exposure to build up those memory cells, so that later on down the track, if a really big response is needed, they can do it. All right, let's talk about some types of pathogens. Viruses are one of these. They are submicroscopic. They are made of DNA and RNA inside of a big protein coating. They are not alive, so they have no metabolism. Because of this, they can't survive on their own. And what they do is they invade the host cells, and they kind of piggyback on that cell's ability to transcribe and translate DNA, and it makes copies of itself. So there's a really sneaky way to infiltrate a host cell and get it to make your DNA. So viruses invade a variety of cell types, but each virus has their specific favorite cell to invade, essentially. Now, HIV is the human immunodeficiency virus, and this is a really important and quite scary virus that appeared in the human species. Either way. All right, so what this does, the HIV virus essentially invades specific immune cells called T helper cells. Now, this is particularly cunning because these cells are there to detect foreign invaders and activate the B cells to make the antibodies. So if you hide within a T cell, the virus can go completely undetected by the immune system for really long periods of time, all the while it's making copies of itself so it can go out and invade more cells. So once the virus copies itself, it can destroy the host cell entirely so that all of that RNA and DNA is released into the plasma, and then it can go and invade other T cells. Now, this means that we are decreasing how many T cells we have, and it completely compromises the immune system. If we don't have many T cells, the B cells really can't be activated as much as well. So that means that we've got decrease in antibodies being produced, which means we can increase how many infections we pick up. And these are called opportunistic infections. HIV positive people have so many opportunistic infections because they are immunocompromised, right? With more and more of these infections, complications can arise, and that's when we get the acquired immunodeficiency syndrome, or AIDS as you know it. Now, HIV is transmitted via bodily fluids such as blood. It can be transferred via breastfeeding, intravenous drug use if there's blood there on that needle, sexual intercourse, and it's minimized by having safe sex, using condoms, all those kinds of things. It is not transmitted through spitting, hugging, sneezing, coughing, kissing, all those kinds of things. So there's so many myths and stigmas associated with this virus. Currently, no cure, all right? No currently available vaccine either. There are management treatments, right? But that's about it. There's preventative measures as well. Now, it is one of, if not the biggest cause of death due to infectious agents across Africa, and it's generally considered chronic rather than acutely fatal in most parts of the world. So it still exists, but it's not necessarily considered fatal in the developed nations. All right, let's talk about different types of pathogens. These ones are bacteria. So we can assist our immune system by treating bacterial pathogens with antibiotics, and these kill bacteria, or they might just inhibit their growth. Now, what they do this by blocking prokaryotic processes. Bacteria are prokaryotes, and they don't actually harm eukaryotic cells. So this is a really good way of being able to kill off an infection and not actually hurt us. So the way they can do that is to include really specific ways that they can stop replication of DNA or RNA or transcription, translation. They might actually stop the ribosome function, the cell wall formation, things like that. But they are not effective on viruses. We know that viruses are not alive, so that's not going to be very helpful. Now, these are naturally occurring compounds. They were initially found in fungi, which was able to compete, I guess, for nutrients because they're both looking for dead organic matter, bacteria and fungi. So they are immobile, fungus, so they want to kill off their competition, essentially. So they do that by secreting a bacteriocide, so something that is going to kill off the bacteria in the near vicinity. And Penicillin is a fungi that was initially discovered to be the first producer, or by accident, really. So, as I said, penicillin was discovered by accident in 1928 by this guy, Alexander Fleming. He was growing bacteria on an agar plate and it became contaminated. So the fungus shouldn't have been there, but it grew there. What he found was there was no bacterial growth in a halo around that fungus. Now, this guy, sadly, was a pretty poor communicator, like many scientists, and his findings were kind of ignored. It took until 1940 for an Australian, Howard Florey, this guy right here, and his partner Ernest Chain and his team to demonstrate that there is a medical application to this. What they did was they injected 8 mice with fatal bacteria, and they gave 4 of them penicillin as well. So the 4 with the penicillin survived, the other 4 unfortunately died. Now, they proved that this is harmful to the bacteria but not harmful to the host organism. We are eukaryotes. They got a Nobel Prize for Medicine in 1945 for this. So antibiotics such as penicillin were analysed for their chemical structure, and they made synthetic versions of them so that the chemical stability was a bit better, and the reliability of supply and all those kinds of things. Funnily enough, bacteria have developed and evolved ways in which they can transfer genes, and those genes, stop them being able to be susceptible to these antibiotics. So it might stop it from entering their cell wall, and it can allow them to become resistant or immune. Now, bacterial populations can become resistant in really, really quick time frames, okay? They reproduce very quickly, so their new generations pop up very quickly. So passing those genes on is not very difficult for them. We are starting to get incredible amounts of resistance in bacterial strains. You might see this hanging on my book bag. This is methicillin-resistant Staph aureus, or sometimes known as multiple-resistant Staph aureus, because it is exactly that. It is resistant to many, many strong versions of antibiotics that we have, and sometimes you'll hear them being called superbug, and that's why it's wearing a cape. All right, antibiotics, we are trying to really prevent this antibiotic resistance because we haven't actually found many new versions of antibiotics in the last 30-ish years, right? So we've got to stop over-prescribing them. They should really only be used for serious infections only, and most certainly not for viral infections. So sometimes doctors give them out just because a patient feels like they need to have something. So completing an entire course of the antibiotics is really important to completely kill the bacterial population and not allow any resistant bacteria bacteria to continue to multiply. Really good hygiene practices need to be happening, especially in hospitals where you can actually go in with one thing and come out with a very resistant infection. Not very good. And they're trying to decrease the amount of antibiotics are used in livestock feed. They are sometimes put in there just as a bit of a protective measure to keep their herd safe. We really do need to find new antibacterials though. All right, here are our syllabus dot points. Remember to watch both videos, please.